But we'll start today with a presentation from Derek Wright. So Derek has a BSc from the University of Regina and a master's in agrobiotechnology from the University of Gießen in Germany. And he now works in the Department of Plant Sciences at the University of Saskatchewan and has been involved in 3 large research projects: the Agile project, Evolves, and then PERC, all working with lentil. So in the talk today, Derek will discuss dissecting lentil growth across multi-environment trials with UAV imaging. So welcome, Derek.
That's the next slide.
Yeah, that works. And then there's some on.
Testing. Good. All right, welcome to my presentation, Dissecting Lentil Crop Growth Across Multi-Environment Trials Using Unoccupied Aerial Vehicles. For those of you that aren't familiar with lentils, on the left I have a photo of lentil plant from the greenhouse, along with another photo just kind of showcasing some of the cool seed diversity that we have within lentils. For us, lentils are a very important crop because we are the world's largest producer now. In 2019, Saskatchewan produced almost 40% of global lentil production. As a result, one of the things we did was to create a lentil diversity panel, which is a big population of 324 lentil varieties, theoretically representing global genetic diversity in lentils, which I've tried to illustrate with this plot, just kind of showing you the origins of all of these lentil varieties. As part of another research project, we had grown this big population in 9 locations across the 3 major macro environments for lentils, and in 2 of these macro environments we were able to have drones fly over our field trials to collect data for us. One of them was in Metaponto, Italy, and then the other one was right here in Saskatchewan, where we had 2 locations in Rosthern, Saskatchewan and Sutherland, Saskatchewan. For Metaponto, Italy, we had 1 site year of data. Rosthern, we had 1 site year of data. And then in Sutherland, we had 2 site years of data. So in total, 4 site years of data that we have drone images for. On the bottom here is just a drone image of one of our field trials. And then up top is just a kind of a photo of 2 contrasting lentil plots just to show the diversity in crop growth that we have within this population. Another thing that we need to keep in mind is genotype by environment interactions. So for Italy, lentils are normally planted in the fall. So in our case, it was November 28th. And as a result, lentils there are emerging into very cold or relatively cooler temperatures with low photo period. And then as a result, there's a large delay in growth and flowering. Whereas in our environments in Saskatchewan, we're planting in the spring, so May 4th in our case. And as you can see, our lentils in our case are emerging into higher temperatures and very high photo period, which then accelerates growth and flowering. So in our case, our whole 324 lentil varieties will flower and mature within 100 days, whereas in Italy, that same population, it'll take 100 days just for the first plant to flower. So our goal is to try and utilize drone data from all of these locations to help us kind of dissect some of the different growth strategies for lentil in not only a large diverse population, but also in contrasting environments. So the first thing we do is we take those big orthomosaics, plug it into a program developed here at the university called Plot Vision, segment all of the individual plots. Once we've done all this plot segmentation, we can then extract data from these images. And in our case, we are interested in things like volume, height, and area. And not only that, but we have data throughout the field trial. So in our case, DAP stands for days after planting. And so for this trial in Italy, we were able to get data on day 94, 100, 118, 132, and 164. Here in Saskatchewan, in our locations, Rosthern and Sutherland, we're able to fly a little bit more frequently, so we have more data. And all I really want you to take away from these graphs is just the sheer amount of data that we're getting. Each one of these little blobs is going to represent 324 data points. And the reason for that is because the alternative is to go out and manually collect this data, and that is a very tiresome and time-consuming process. So in this photo, you can see two people that are manually collecting measurements for canopy height, yelling that data out to me, who's on the tablet, plugging it in. And we tried to improve this a little bit by taping barcodes to meter sticks and then using barcode readers. But still, this is a very time-consuming process when you have thousands of plots. So then ultimately, once we do get all this data from the drones, first thing we need to do is ask ourselves whether or not the data is very good. So in this case, we have on the y-axis is canopy height measurements from the drone correlated with our manual measurements of canopy height, and this is across all 4 of our site years. In the bottom panel is measurements for plot volume from the drone correlated with our manual measurements of biomass. And as you can see here, we do have some pretty good correlations coming out of the drone, with the exception of canopy height in Italy. However, I won't go into the details of why that might be. After that, the next thing we can do is, what we want to do is try and calculate growth curves, because we do have data throughout the experiment. So here I've just kind of shown a couple different plots just to see that progress over time. So here is the modeling of growth curves that I've done. Now this is just one variety. In the top panel is, I have, the axis is cut off there, but so in the top panel is modeled growth curves for plot volume for just one of the 324 varieties, and then it's across all 3 reps for that location. In the bottom panel is the same thing, but for Rosthern, Saskatchewan, as opposed to Italy. And then I have drawn this kind of vertical green line just to indicate when they're flowering. So what we can see is for this one line, in Italy, it's doing all of its growth for volume and then flowering, whereas in our environment, the flowering is kind of occurring right in the middle of growth. Now, in comparison, here's another variety just to contrast with, and you can see for this one, not only is it much smaller in volume in both environments, but also it's flowering prior to its growth. So just to illustrate like a very different growth strategy that we have here, and these are just 2 of the varieties. In addition to that, we can also do growth curves for height and area, and so here I have growth curves for height in Italy, height in Sutherland, Saskatchewan, area in Italy, area in Sutherland, Saskatchewan, and I've plotted all 324 of our lentil varieties here, and then I've just highlighted 2 of them. And so you can see for this, the green line, for this variety, it has very high height, high area in both locations, and then the red line has low height, low area in both locations. So this is just kind of nicely illustrating a genetic component to growth. If we then take a look at 2 different varieties, now we can see for these ones, if we just focus in on Sutherland, Saskatchewan, we can see both of these kind of have roughly similar patterns for both height and area. However, once we go over and take these 2 to Italy now, we can see that one of them has relatively high height and low area, and the other one has relatively high area and low height. And so this is just kind of nicely illustrating a very strong G by E interaction going on. Another cool thing we can do with these growth curves is kind of extract some additional information and data points from these growth curves. And so I've tried to illustrate that with this little figure here. So some of the things we can do is figure out, okay, what is the day at which the midpoint for growth occurs? What is the day where you get the maximum growth? How long does it take for the doubling time of this line? What is the growth rate during this linear portion of the growth? And then I've also have this green line again to indicate when they're flowering. So now we can ask ourselves, when is all this occurring in relation to their flowering? And so these are just extra data points we can extract out of that. Once we have all of this information, another thing we can do is do a principal component analysis. And by doing some clustering, then this is where we can then try and dissect some of the different growth strategies within this diverse population. And I don't have time to go into this in more detail. However, I do have a poster. So if you are interested in knowing some of the more nitty-gritty details after this, come see me in my poster and we can talk in person. And then I just want to finish off by pointing out that our next step is going to be to take all of this data and run genome-wide association studies to then get at the underlying genetics behind all of that. And with that said, I just want to thank all of the appropriate people, and I'll be happy to take any questions. And just to also reinforce, I do have a poster, so you can also come talk to me and ask me questions there as well.